Actually, Bing, do you want to share your slides?
Okay, let me share my screen. Can you see it?
Yes, perfect. Great. So let's get started today. Welcome to today's Immune Zoom seminar. We have a treat today. It's my great honor to introduce today's speaker, Dr. Bing Su, who is currently the director of Shanghai Institute of Immunology and the chair of the Department of Immunology and Microbiology at Shanghai Jiaotong University. Bing did his undergraduate in Peking University and a PhD at Yale. He did his postdoc training with Dr. MEKK at UCSD before starting his own group at MD Anderson in 1995. He was promoted to tenured professor there. In 2007, Bing moved to Yale Immunobiology. And then Bing joined the Shanghai Jiaotong University in 2014. Bing's research focuses on signal transduction and immune regulation in both healthy and pathological conditions. He has made numerous groundbreaking discoveries on understanding how mTOR and MAPK pathways regulate immune responses, metabolism, and vascular formation. For example, Bing has recently published a beautiful Nature paper revealing a novel subset of intestinal stroma cells that protect the intestine from acute damage. He identified a molecular pathway in this cell type, starting from ROS and ending in production of R-spondin-1, which enhance Wnt signaling and promote tissue regeneration after injury. So this is indeed really beautiful work. Bing has authored over 130 peer-reviewed articles. He also served on many review committees, including NIH and the NSFC. Bing is an enthusiastic educator and has done extensive teaching at multiple universities in US and China. Importantly to mention, Bing took efforts to promote international collaborations, not only for his own group, but also at a higher level. He initiated and co-directed the Shanghai Jiaotong Yale Immune Metabolism Institute. And he has also organized many international symposia to facilitate scientific communications across the globe. I think this is extremely important, especially during this pandemic era. So without further ado, let's welcome Bing.
Okay, can you hear me well, Shelley?
It's great.
Can you hear me?
Yes, it's great.
Okay, all right. Okay, first I would like to take this opportunity to thank Wu Chuan for really the great effort to organizing this really truly wonderful online seminar series, the Immunozoom. As many of us already hear from many great immunologists presented our researches at this forum. And also I want to thank Xiaolei. Xiaolei 是我的本家，算我的小弟。So I hope you were not confused because Xiaolei gave a talk last week and did a favor for me because I was traveling, and so he gratefully agreed to switch his slots with me. So we both switched the slots, and we also all had a Yale connection. So maybe you know, it will confuse some people with Xiaolei. But indeed, as Xiaolei just mentioned, I spend quite a lot of time at Yale, both as a student as well as a faculty, and I still have a house there. and or it's missing the life in New Haven and the pizza, as Shanghai just mentioned. Our work today I would like to present actually did not include what Shanghai just mentioned. We recently published Nature about the stroma cells. Rather, I wanted to really share with you one of the areas that in my lab has been working since I get independent. And this is really a work carried on from the lab where I did my postdoctoral training, Dr. McCarran's lab. So I worked on the kinase called MEKK2 and 3, which is an upstream activator for the mitogen activated protein kinase cascade. As you may all are familiar with the eukaryotic MAP kinase cascades, that's including the well-known ERK1, 2, GNCase, P38, and ERK5. Those are the main MAP kinases in eukaryotic cells. And this kinase cascade normally involving 3 kinases. At the bottom is MAP kinase, which is activated directly by its upstream kinase called MAP kinase or MAPK2. And the MAPK2 in turn is activated by its kinase called a MAPK2 kinase, or in short, a MAP3 And the MAP kinase cascade is highly conserved, and you can find a very similar kinase cascade in single eukaryotic cell, such as yeast, all the way to mammal and humans. And they are able to respond to wide spectrum of upstream signals, both intracellular and I would say both extracellular and also intracellular as well. So most of the started signals, including growth factors, environmental stresses, inflammatory cytokines, such as the pro-inflammatory cytokine R1, TNF, etc., and also tissue factors such as angiopoietins, as well as many developmental signals to drive, the tissue and organ development. And through the cascade, MAP kinase can modulate a lot of substrates, including membrane proteins, cytosolic proteins, ER proteins, and the nuclear proteins, including transcriptional factors. And those, as you know, can finally, lead to cellular differentiation, proliferation, development, or inflammation or inflammatory cytokine release, such as. So it is known the MAP kinase, the cascade, is very important, but now we know all the genes, the pathways, but still there's a lot of things are unknown. So my lab, since I started my lab at the MD Anderson Cancer Center at Houston, I studied 2 kinases called MEKK2 and MEKK3, which I initially cloned at Michael Karen's lab. And when I started my own laboratories, I focused on these 2 kinases. As you know, it belongs to the MEKK11 gene family, and it at the catalytic domain share quite a high homology with East STAT11, which is East MEKK. And so far, at least there's 6 members of MEKK are considered in these families, but most known the MEKK1 has a very long side N-terminal sequence of the catalytic domain. Well, MEKK2 and 3 are relatively smaller as compared to MEKK1, and they are very similar. These 2 kinases, as you can see, at the kinase domain, they share almost 98% of identity, and it's N-terminal regulatory regions also share around 70% of homology. And both of them are very strong activator of C-jun N-terminal kinases. And it can regulate phosphorylate and activate GNKs, but also can phosphorylate other MAPK such as MEKK3 and MEKK6. So these two kinases at the biochemical level are very similar, and they also express broadly. And if you look at the gene structure in human and the mouse, they are very, very similar, indicating these two kinases may evolve from one gene during the evolution, and they must have a shared function, but also specific function. And interestingly, at the early time when we studied these two kinases as well as other MEKKs, we found that when you just ectopically express them in cell lines, you can lead to uncontrolled MAP kinase activation, but they do have a specificity. So if you look at MEKK2 expression together with RAF and the RAS, which they are upstream activator of ERK, those are the kinetic activators for ERK. And MEKK actually can much strongly activate the GNK, okay, but not ERK. So indicating the MEKK has its intrinsic specificity. I'll later on show some of the data to demonstrate that. So it's quite interesting. In addition, many other MAP3 cases such as TAC1, MLK, etc., they also share quite a similar property as if you ectopically express them, they can automatically upregulate the MAP kinase. And in addition, we find that MEKKs can activate IKK and kappa B pathways. Therefore, the function and regulation of MEKK seems much more complicated. Not just as a simple MAP kinase module. So there are two possible ways to change the MEKK2 signaling in vivo using the model system, as well as probably you can find in some diseases. That is, if you delete or knock out either of those genes, MEKK2 and MEKK3, then you would have a complete loss of functions of MEKKs. But on the other hand, the MEKK in the cell can be abnormally activated either by slightly overexpression or by inactivation of the inhibitors, which we know in the cells. They are controlled by inhibitors, as I mentioned, why it is ectopically expression would lead to uncontrolled MAP kinase activation, because there's an inhibitor titrating MEKKs in the cell. If inhibitor is lost, then you also would upregulate MAP kinase or its specific downstream MAP kinase activities. So we look at the expression patterns of MEKK2, for instance. Here you can see in almost all the tissues in the cells, you can find the expression, although some tissues show slightly more expression, some are less, but you can clearly see they're very well expressed, either in the tissue, including lymphoid tissues like lymph node, spleen, thymus, but also in the purified immune cells such as T cell, CD4, CD8, B cells, or epithelial cells as well, in both intestinal epithelial cells. So both MEKK2 and 3 are very well expressed, and in many cases, they are exposed in the same cells. And we, very early on, we made a knockout mice, but at the same time, we're knocking the Z gene into the MEKK2 locus, as you can see. As early as embryonic day 10, you already start to see some expressions of MEKK2, and those expressions are gradually increased into multiple tissue and organs as the embryo develops. So this, really indicating these two kinases as a very important roles in either development or in the early functions. As I mentioned, each MAPK module has some specificities. So early on, we know MEKK2, JNKK2, JNK1 can form a very specific tripartite complex that allow a specific signal to mediate through the JNK pathways. We believe MEKK3 also has a very similar mechanisms. And in addition, we have identified an MEKK3 regulatory phosphorylation site. As you know, just like other MAP kinases, we know there's a conserved TPY motif in the kinase domain, which needs to be phosphorylated by MAPK2. But in the MAP3K, it's been argued, whether they have such a regulatory phosphorylation site. We indeed identified a serine 519 in MEKK2, and at the same position in MEKK3 is serine 526, and that is phosphorylated by upstream signals. I would tell you it is not by upstream kinases. Indeed, it is autophosphorylation by itself. If we mutate this site, you can see there's a dramatic reduction of its activity towards JNK and also JNKK2. So indicating this activation is very important. And we generating the phosphor antibody specifically can detecting this phosphorylation, as you can see. If we made a kinase dead, they're not phosphorylated. If we mutated this 519 to adenine, there's not phosphorylated. But wild type truncated or the adjacent threading mutations did not affect this phosphorylation. So we had a very nice, very specific phosphorylation specific antibody to detect the MEKK2 activity. Indeed, utilize this antibody, we demonstrated that MEKK2 can be activated by multiple upstream cues, including pro-inflammatory cytokines such as IL-1, TNF, also by growth factors such as EGF, or by toll-like receptor ligands such as PGN, LTA, CPG, or by stress such as UV or sorbitol, as you know, which is a very strong upstream activators of P38 pathways. So this indicating MEKK3 or 2 activation requires phosphorylation. And this phosphorylation is utilized by a self-dimerization formed protein-protein interaction leads to self-phosphorylation. So we have also proved that this self-interaction is indeed required for both MEKK2 and MEKK3 activation for the downstream activation signals. And this also led us to believe why topically expressed MEKK2 would automatically lead to the downstream MAP kinase activation, because you increase the concentrations of MEKK2 and MEKK3, and either the tethering molecule or the inhibitors of MEKK2 and MEKK3 cannot prevent this self interaction step and activation, therefore leads to the downstream pathways. And early on, we showed that if you are titrating the MEKK2 expression, you can synergize with CD3 stimulation or TCR signal to lead to the downstream JNK activities you can see. And this is different from the under CD28 stimulation. So indicating MEKK2 would work downstream of TCR signal in activating T cells. Indeed, if we utilize a dominant negative MEKK2, and we can block in the T cell growth factor expression IL-2 using IL-2 reporter, you can see a very nice dose-dependent effect of inhibition. And of course, with no surprise, it blocks CD3, CD28 induced AP1 activation by dominant MEKK2 molecules. But to us, is when we knocked out MEKK2 in vivo by gene ablation, And we do not find MEKK2 deletion affect T cell differentiation, for instance. In vivo, we do not find any problem with the tissue residential T cells, such as the cells in the gut. We find that both the Th1, Th2, Th17, and the T regulatory cells, they all are compatible. And data not shown here. We also find that T cell proliferation, dynamic T cell development, all the regular things you check with the T cells seems not affected, except we find if we utilize CD3, CD28 to stimulate naive T cells from the differentiated mice, and they give increased response both at the proliferation, which early time we use treating incorporation, or by looking at cytokine production of out and interferon gamma, indicating that although MEKK2 is not required for T cell response, but somehow they may have some negative regulatory roles that can balance the TCR CD3 signal to not allow an exaggerated T cell activation, and the deletion of it possibly can be compensated by MEKK3 to transduce the signal, but maybe the missing some negative regulatory functions, which we still don't know. But we do know that when GNK is very much activated, that normally would phosphorylate MEKK2 to downregulate its activity, and possibly MEKK2 can do something to further downstream downregulate upstream signals when the whole pathway is activated. But this is still a question to be investigated. But is there a tissue-specific role of MEKK2 in T cells? So we did this rack transfer experiment, as you know, that if you put naive T cells into rack mice, that will induce colitis in the recipient mice due to the lack of T regulatory cells in the recipient mice. But when we compare the wild type and MEKK2 knockout T cells in these models, we found it very interesting. The MEKK2 knockout mice is unable to induce colitis, indicating there's some deficiency as compared to the wild type cells. We did all the reasonable experiment to prove this is true. And what we really found is that when MEKK2 is deleted in this model, Th1 cell is reduced, but the Th17 cell seems increased. And therefore, we believe the increased colitis, which has been also published by many other groups, is due to the increased Th1 cells in this right intestines. But in this case, even we do see an increased Th17 cells in our model in the knockout mice, they actually also do not cause diseases. So we believe the main effector cells is the differentiation of naive cells into Th1 cells. So to look at that, we find that the pathways that are being affected is not ERK or PC-related, but as you can see clearly, the GNK activation is reduced. In fact, we can use GNK inhibitor to mimic MEKK2 deficiency in this model, indicating that the MEKK mediated role of Th1 differentiation is dependent on GNK. And what is the triggers for this differentiation? We tested a number of factors that is known to affect Th1 differentiation, but we found only IL-18, as you can see, that IL-18, when given to the cells, they will promote the Th1 proliferation differentiation. But this increase was blinded in MEKK2 deficiency, indicating that the IL-18 promoted Th1 cell differentiation in the RAG1 deficient mice is mediated by MEKK2. So this is the models that we propose in the differentiation of Th1 cells, which is required for the colitis development in the RAG deficient mice, can be further enhanced by IL-18 in the gut tissues. But this signal requires MEKK2 to drive these Th1 cells to lead to the colitis development. And this actually was recently published How about MEKK3? MEKK3 actually was one of the early kinases. We started this one more earlier than MEKK2 because we found it has a very nice vascular phenotype. We actually did a lot of work when I was at MD Anderson and also at Yale. Particularly when I was at Yale, I was at the vascular and therapeutic programs on this. And it has a very prominent vascular phenotypes, both in the brain, which is originally proposed because it is associated with one of the brain vascular diseases called CCM, called cerebral cavernous malformation, CCM. But we also shown that MEKK3 regulates the vasculature in many other places, including lung, kidney, and intestine as well. And it has a very different role when we knock out MEKK3 as compared with the diseases in the CCM, which I'll mention later on. In the CCM, actually, the disease is caused by over MEKK3 activation rather than the MEKK3 deletion. So this is probably due to totally different mechanism, which we are still trying to understand it. But when we had the mice, we isolated embryonic fibroblast to study the pro-inflammatory cytokine signals. So early on, we really demonstrated that it is required for the IL-1 receptor for the TLR receptor signal, and that's mediating the GNK, ERK, P38, as well as IKK activation. And those work are published early on in Nature Immunology. So if you're interested, you can take a look of those ones. But here I wanted to further to show you some of the data on the role of MEKK3 in T cells as compared to MEKK2 knockout. Again, in the thymus, we do not see much difference of MEKK3 deficiency. This is utilized both by CD3-CRE and LCK-CRE. But also, there's no problem with the thymocyte activation and survival after MEKK3 deficiency. However, we do find a phenotype as compared with MEKK2 knockout, indicating this is MEKK3 specific function, because in these cells, MEKK2 is also expressed. And you see the peripheral spleen or lymph node, you have both reduced the CD4 and the CD8 T cells. And this is a lymph node as well. So we spend quite a lot of effort trying to figure out what is the defect. We did this well-known mixed transfer experiment by transfer the spleen T cells with either the congenital white type cells into a recipient mice, which we can use a surface marker to separate them. As you can see, in the same host, the knockout is unable to compete with the white type T cells to proliferate, both in the proliferations, particularly this group of so-called slow proliferating cells, but not the highly proliferating cells, which are probably stimulated by superantigens or other stimuli. Indeed, when we take the naive cells for MEKK3 knockout host, and give to strong antigen stimulation, they actually proliferating quite similar. So indicating this defect is only associated with the phenotype called T cell homeostasis. That is not antigen driven, but self-antigen and also common gamma chain cytokine driven events. And we tested the IL-7 response, and we do not see any difference between wild type and MEKK3 knockout CD4 cells. This indicating that MEKK3 knockout T cells, when we look in the T cells from this mixed transfer, clearly we see a deficiency of ERK1/2, but not P38 activation in this lymphopenic host. when we do the mixed transfer. And this leads us to believe MEKK3 is required for this self-antigen stimulated homeostasis. And if you mix wild type and MEKK3 knockout naive T cells into the host, because it had a reduced T cell signal as compared with the wild type T cells, therefore they cannot proliferate as well as the wild type T cells. Therefore, you would see this phenotype that you had a very dramatic reduction. When you started with 50%, and now you only had like 13% of wild type cells as compared to the co-transferred naive T cells. So this is one of the unique function of MEKK3 in T cells, we believe. But as I said, these two cells can compensate for each other. Therefore, we do look for the double knockout of MEKK2 and MEKK3 in CD4 T cells. And this is work done quite some time ago by my former postdoc, Can Xin. He's now a professor at Westlake University. And as you can see, that when we knock out both MEKK2 and MEKK3 in CD4 cells, they have an accumulation of this so-called activated memory-like cells, which is CD44 high and CD62L low cells. Most interestingly, we find there's a deficiency of MEKK2, 3, ERK1-2 signal mediated SMAD2 and 3 linker phosphorylation. Okay, and this phosphorylation will lead to lower SMAD transcriptional activity. Therefore, if you knock out MEKK2 and 3, therefore this increase the linker phosphorylation will be reduced or lost. Therefore, you have more SMAD2 and 3 activation, right? And it is known these are very important for both Th1 and Treg cell differentiation in vitro. And indeed, we find in the double knockout mice, you can see there's a very dramatic increase of both T regulatory cells as well as Th17 cells. But there's a, the same time you see a little bit reduced the Th1 cells. And this indicating MEKK2 and 3 can, through the TGF-beta signal pathway, to modulating the Treg cell and the Th17 cells. We'll also utilize the EAE model to demonstrate the increase the Th17 cell is indeed pathogenic in the EAE mouse models. Therefore, MEKK2, 3 would be important to balance the peripheral effector T cell homeostasis and activation to prevent the such autoimmune diseases as EAE. So I don't like these slides, and I just recently trying to organize a little bit about the work we have looked at the role of MEKK3 in the immune cells that some of them I discussed, but some of them I did not mention, such as the vascular systems through ERK5, KLF, and the vascular structure and the tissue repair. What I wanted to really to show here is one of the partners of MEKK2 called CCM2 and CCM2L. As I mentioned, this is a brain vascular diseases, right? And they've been known to be a negative regulator of MEKK2 and MEKK3. So in this CCM diseases, which is a loss of function of CCM1, CCM2, or CCM3, and they will lead to the vasculature abnormality, has this dilated venules in the brain, and in the severe situation will cause the disruption, so-called hemorrhage in the brain, and in the really mild diseases will cause very bad headache. So the genetic link are the 3 CCM genes, CCM1, 2, and 3. But we found that MEKK3 only strongly interact with MEKK2. but very little or no interactions with MEKK, with the CCM1 or CCM3. So indicating there's a very strong protein-protein interaction and a functional interaction between MEKK2 and MEKK3. And indeed, we also did a very similar experiment and identified the residues of MEKK2 and MEKK3 interaction at its N-terminal sequence of MEKK3. And we also show MEKK2 can interacting with CCM2. So this really led us to believe, by looking the vascular studies showing that CCM2 can inhibit MEKK3 normally. If this inhibition is lost, then you would develop CCM pathology. So overactivation of MEKK3 actually is not good for the cell, particularly for the vascular cell. And we found that very, very importantly, in immune cells, particularly in T cells, you have a very high levels of CCM2 and the CCM2L expression, okay? As I mentioned, we showed the phenotype of MEKK2 and MEKK3 in the T cells. So how MEKK2 and MEKK3 is regulated in T cells in responding to upstream signals that is involving involving CCM2 and CCM2L is currently unknown. Now we are generating many reagents, including the specific antibodies, locking tagged CCMs, as well as conditional knockout of CCM mice is trying to build these models to further working on the mechanistic side of MEKK3 regulations of T cell functions. So what we're recently testing is how MEKK3 may regulate CD8 T cell responses while controlling T cell signal strength in chronic viral infection or in cancer. And then we wanted to know how this pathway MEKK3 through the KLF, which we already identified both in vascular system, in the stroma cell systems. They also may regulating this CX3CR1 positive effector T cell differentiation and migration. These are the very important anti-tumor CD8 T cells. And we are looking, using single cell RNA sequencing data from our own lab as well as in the database, trying to, to really identify the additional modulators such as CCM2, CCM2L on the MEKK2 and 3 in the T cells. And we did some very preliminary data by inoculating colon cancer tumor cells, MC38 tumors, onto the double knockout mice as compared to the flux mice, which is the control mice. As you can see, there's a much bigger tumors as compared with the control mice, therefore indicating maybe there's a immunodeficiency of this double knockout mice and the tumor activity. Indeed, when we gated on the tumor infiltrating CD8 T cells, we found there's a great reduction of this CX3CR1 positive, PD1 positive CD8 T cells. You can see here, those are the effector cells against the tumor. So if you have reduced these cells, of course you would see a reduced the tumor immunity. And at the same time, we see a two subset of cells which are more probably like exhausting CD8 T cells in the tumor that are increased as compared with knockout mice. So indicating indeed the MEKK2 and 3 may have a role in the anti-tumor CD8 T cell function or maintenance for the function. So we are trying to, really expanding these areas to looking the longevity and looking for the exhaustion of CD8 cells in chronic and viral or anti-tumor immunity, and also looking into the mechanistic side either involving this CCM2 and the CCM2L molecules. So I would like to stop here to acknowledge many people, including former lab members that really started the whole MEKK project. That's including my first postdoctor, also my technician and visiting scholar, my classmate in Beida, and Jin Ke-chen, my first postdoctor fellow in my lab to start in my lab at Anderson Cancer Center. My other postdoctors, Chao Jiahuan, Kiwan King, etc. So I'm not going to name, but also I'm very happy that MEKK2-3 project is continued in Shanghai by my students here, my current lab members, including Ningbo Wu, Song Xiang, Tan Jianmei, etc. And particularly, I'm happy that the T-cell part will be continued by Chen Yao. Actually, she did her PhD with Cui Weiguo, and he also given the immunosome presentation a few weeks ago, and with the Yale connection, we got trained with the SUCAC at Yale when I was a faculty there, and also by my incoming graduate student Xu Ziyang, and of course without collaborations with many good friends and collaborators at in US and in China. This work cannot be done. So I'll mention some of them, like Liu Zhenggang at NIH, and many at MD Anderson, including Chen Dong when we were colleagues there, and also in UT Southwestern, like James Chen at the early time when we studied the IKK and MEKK3 connection. At Duke University, Yuan Zhuang, I actually had a very happy news to tell you. Yuan Zhuang was had a problem with collaboration with China, and Duke just reinstalled him so he can go back to restart his research at Duke. So I'm very happy to hear this news this morning. And also in Houston, I wanted to acknowledge my friends Qin Jun and also Puming at the Baylor College of Medicine. And of course at Yale, there's many friends, including recently, of course, I probably will do more with the Xiaolei, hopefully, okay, including here, but in the Yale SUCAC, and Richard and Dan and Isong now at UNC, etc. give me a lot of help. And currently we're working with our affiliated hospital in Shanghai, including Region Hospital, etc. So I would like to stop here and really again to thank Chuan, Wu Chuan and Xiaolei for both of you for your kind invitation and hosting me today here. Thank you very much.